So this follows on, I think, nicely, but as a clinician, what I see in clinic is a disease that's currently incurable, and we'd like to change that. And actually, one of the things I want to propose really is that we should be looking at very early disease, and ideally that sort of point between preclinical and clinical disease. So the way I look at it, patients are born with some kind of susceptibility, which is determined by their genetic profile and perhaps something that happens in development. And then they go through a period when they certainly don't go anywhere near a neurologist, but there's something going wrong in their system. I don't know how long it lasts for, but there's a compensation, and that's the preclinical phase, and it could last for decades, or it could last for months, we don't know. But by the time they present to a clinic, they've already had the disease on average for 1 year, and what we're really looking at here is the system in decompensation, and trying to find treatments that really alter that have proved very challenging, and particularly when we're doing current trials in this phase here. So that's something we need to change if we really want to make progress. So the way we're approaching this is to try and focus on early disease both in human beings. So we're looking certainly at patients who present to our clinic as early as possible, but then we're ascertaining if they've got C9orf72 mutations, they're asymptomatic relatives who are carrying the mutation, and we're doing the same thing for SOD1 in collaboration with Michael Benatar in the US. And that's our sort of systems-based approach using imaging and wet biomarkers. The mouse models I'm going to tell you about in a second are models, I think, potentially of early disease as well, and then we're trying to validate that in human iPS-derived motor neurons to try and create some sort of triangulation. That means we can really understand which are the relevant changes. That's the aspiration, anyway. And of course, there's a terrible history of 20 years of looking at this poor creature, which has had high levels of expression of protein, which has been derived from a cDNA, concatenated into the mouse genome somewhere, and driven from heterologous promoters to very high level with protein of 10 to 12-fold elevation, and it dies in its juvenile period. And you can really find things that are going wrong very early on. The earlier you look, you can see that really almost from birth, things are going wrong in this mouse. So it's got probably some developmental abnormalities. And people have focused very heavily on whether the mouse dies or lives, which I think is a problem for therapeutics, really, and a poor readout of what might work. And curiously, the FDA still require companies, if they have a candidate they want to take into clinical trial or take to market, to show that they've used it in this mouse model as the best established model that's out there. So it's actually causing all sorts of problems, I think. We may be discarding drugs that actually might work, for example. And because here's TDP-43 in the nucleus of the SAD mouse, and there's no TDP-43 pathology at all, so clearly it's potentially an outlier in the context of a disease as a whole. So obviously TDP-43 is a proteinopathy, ALS is a TDP-43 proteinopathy, and the reason I show this slide is simply that it seems to me that the pathological hallmark we need to be looking for in models is, and these are 3 motor neurons from the adjacent region of one segment of a spinal cord of a patient who died, and there are motor neurons where TDP-43 is in the nucleus, those in which it's gone into the cytoplasm in a speckled pattern, and those in which it's aggregated, all very close to each other. So that's possibly the hallmark we need to be looking for to understand why the disease occurs. And we know, and many people have made the point already, that overexpressing TDP-43 is a toxic thing to do. So that's something we'd like to avoid. And here, for example, is work from Jean-Pierre Julien using a sort of similar approach to ours, although there are some differences. And actually what they found really is that if you express TDP-43 even 2 to 3-fold higher than physiological levels, you actually get as much problem with the wild type as you do with some of the mutations. So overexpression doesn't necessarily tell you exactly why the disease is occurring. So the ideal mouse model, it seems to me, is one where you're avoiding overexpression or significant knockdown. You have a mouse that's developed a normal neuromuscular system, so that you're not looking at developmental abnormalities, where you can recapitulate that pathological signature I showed you. It should really have something wrong with its neuromuscular system, I think, if we're dealing with ALS. It would be nice if the wild type human TDP-43 expression was normal, but of course, in sporadic ALS, we see endogenous, non-mutated TDP going wrong. So I'm open to the possibility that we might see a phenotype in those mice as well. It would be nice if it had cognitive deficits, so we can study FTD as well, but I have reservations about the mouse as a model for FTD. And reduced lifespan, I guess, would be helpful, if not just to convince reviewers that we're dealing with a nasty disease here. But for me, in terms of understanding the biology, I'm less interested in that. So to address this kind of problem, we created a back transgenic mouse using a system developed by Ben Davis in Oxford, which is an integrase mediated cassette exchange system. So you can basically plug your gene of interest into the RASA26 locus, and knowing that you've got a single copy, and you can put the whole human region surrounding your gene. We've got about 50 kb of TDP-43 here, including all of the exons, all of the introns, the 3' and 5' regulatory regions, and we've shown that that's been cloned intact into this locus in the mouse, both in the wild type and the mutant M337V. And this is just to show you that the level of expression, so it's got a tag, so it actually comes in at 72 kilodaltons, and these are a series of lines with showing that this is compared to endogenous TDP-43 expression. So it's sub-physiological, if you like, although this is obviously coming from two loci. And we show that in the spinal cord of these mice, you get the accumulation of C-terminal fragments in the brain, and all of the sort of pathological signature that you might expect from human brain seems to be recapitulated in these mice with the accumulation of insoluble fragments. This is half speed, and it's a way of phenotyping the mice using something paradoxically called a catwalk. And so we took significant numbers of these mice, male and female, and actually each time the mouse's paw hits the runway, you get all sorts of information. In fact, you get rather too much information, I think. There's a risk of finding things that aren't there, one thinks sometimes. And all of this can be used to understand early changes, because the rotor rod is still a useful thing to do, but tends to occur much later in any kind of natural history. Distal weakness can be tested by hanging wire, and cognitive testing by open field. So although the mutant M337V mice, both hets and homozygous mice, are slightly underweight compared to non-transgenic and wild type, they don't lose weight, certainly up to 9 months, which is really the only time point we have full data. But the homozygous mice are starting to fall off a rotor rod at that point. Grip strength is reduced, both in males and females, so we're getting basically a neuromuscular or a motor phenotype at 9 months. So that fulfills my criteria of wanting the mice to develop normally. We know they're absolutely normal at 3 months and probably normal at 6 months. And there is a dose effect. So at the moment, the hets are not abnormal at 9 months. We've got some early data for 12 months, which is not yet statistically significant, suggesting the hets start to become weak around that time. So pathology. These are not the greatest images in the world, and there's a lot more work to do, but we have TDP-43 in the nucleus in non-transgenic, in heterozygous and homozygous wild-type mice. But in the mutant, we are seeing in cytoplasmic staining for TDP-43 in a sort of speckled pattern. It's not in every cell, but it's statistically highly significant. So this is, again, fulfills the criteria of seeing the right kind of human pathology, and this is at 9 months. It's certainly not present at all at 3 months. So obviously, a lot of work has been done to define the complex role of TDP-43 in RNA handling, but particularly in splicing. And the question that I think is critically important here is this alteration of splicing of the targets of TDP-43, is that abnormal in the disease or not? And I think our model may allow us to look at that, because we can see that there's a period of normality. And if it's mechanistically related to the disease, then you should be seeing splicing abnormalities before the disease phenotype occurs. So as I mentioned just now, if you look at a range of mutants using the splicing assay developed here, then if you knock off the C-terminus of TDP-43, you certainly get a major change in exon 9 inclusion. None of the mutations, they all behave basically the same as wild type. Now, we can't go through all 6,000 targets of TDP-43 to see whether that's abnormal, so we have to do something a bit different. So I'd hoped when this meeting was announced that by now I would have enough data to tell you what the answer is. I'm afraid I don't. So this is just a single time point at 3 months of age where we think this is a pre-symptomatic time point. So if it's mechanistically related, we should be seeing something going wrong. So we certainly see changes in, this is gene expression levels. So there are about 180 genes which are different between the mutant and wild-type mouse. We haven't yet got the analysis for whether there are alterations in splicing, behavior read-through into introns, etc. So that's data that I'm afraid I can't tell you. But this story, is perhaps reminiscent of something I've worked on in the past, which is spinal muscular atrophy. This was a gene that was identified, the survival motor neuron gene. It has a fundamental role in all cells, so that if you knock it out, it's cell lethal. And its role is in the assembly of SM core proteins for splicing and reimportation into the nucleus. So you'd predict that if you had SMN deficiency, which in humans causes spinal muscular atrophy in a dose-dependent way, so you have patients who might be older, who have more SMN around, and you have babies who have very little and they die in infancy, and SMN stains these Gemini of coiled bodies in the nucleus. So the prediction would be that SMN, its known function is generalized in splicing, that if you got rid of it or knocked it down, you would get major problems in splicing. So we looked at that using a mouse model, and we looked at 3 time points. P1, when we know that there are normal numbers of motor neurons, and wild-type and mutants cannot be distinguished. P7, when there's starting to be a separation, and then end-stage disease, to see whether there's any abnormality in splicing. And again, the same principle applies. If this is mechanistically related to motor neuron degeneration, what you would see is a lot of stuff going on here. So when we did that using Affymetrix exon arrays, we found that there's essentially nothing much going on at all at these 2 time points, and there's an awful lot going on here, so that the splicing abnormality seems to be a consequence rather than a cause of motor neuron death here. But interestingly, this kind of analysis allows you to start exploring the early pathological events, as we'd like to do in TDP-43. So we identified that there were chondrolectin is one transcript which is abnormal early on. There were about 10. Chondrolectin is one of them. The reason we got excited about that is that in situ hybridization shows it's exquisitely localized to motor neurons, so that looks like a good candidate for some functionally relevant change. And basically what's happening in the pre-symptomatic mouse is there's an isoform shift from one form of chondrolectin to another. And then with Katerina Becker in Edinburgh last year, we looked at zebrafish to do this, and she knocked down SMN with a morpholino and finds there's an axonal phenotype, just as Bettina was predicting for every other gene. And then if you add in the chondrolectin isoform that is shifted in the humans, in the mouse development, actually you get restoration of this axonal phenotype. So this is simply an example of where these early pathological events might give you some clue as to why motor neurons are vulnerable. And we've already heard that, so the point here is that what you think you know about the function of a gene is all very well, but no gene has one function these days, and particularly in neurons, many of RNA binding proteins have acquired either completely different moonlighting kind of functions, or perhaps more commonly, compartmentalized functions. And so the work on FUS at the neuromuscular junction might be an example, but also this very lovely work here from Paul Taylor showing that TDP-43 is in axons that mutants behave differently to wild type. So these are the kind of changes that we might be expecting to see. So here are some primary motor neurons from E13 embryos, and the M337V mutant, even at rest, when there's no stress at all, is more likely to be cytoplasmically located, suggesting that it's, I guess, behaving in a molecular sense as you might want it to in terms of its compartmentalization. But if you stress the cells, and we've got a lot more work to do here, but normally we would expect, we think that non-transgenic, so endogenous TDP-43 in a time-course stress experiment, that stress granules are formed and that TDP-43 can be found in these stress granules. But basically what we're showing here is that in the presence of the mutant, there is an inhibition of stress granule formation. So whatever the mutant might be doing, one possibility is that it might actually be affecting the cell's ability to cope with stress, which might be a plausible kind of mechanism. But these are rather preliminary sorts of results. And this is just showing that co-localization with 2 markers of stress granules, TIA1 and poly-A binding protein, is impaired in the mutant. The cells can also, we think, be used as a sort of drug screening tool. So in addition to the stress granule formation, there is a slight survival disadvantage, and there's an abnormality of calcium handling, and suggesting the cells are vulnerable to stresses. the whole range of work we're doing showing that ER calcium stores and BCL2 regulation may be abnormal, suggesting that this could be at least a phenotype that you can observe. Whether it's mechanistically related to the disease is another matter. So, in summary, I think that this kind of back transgenesis can lead to a much more controlled kind of mouse experiment where you've got physiological levels of expression, and I guess the concern at the beginning was always going to be, are the mice going to have a phenotype, since previously most people have shown that you need perhaps higher levels of protein. But here we've got not just physiological levels, but we've got intact intronics, and we've got the whole human gene, hopefully allowing it to function in a way that might be what's happening in humans. We've shown that we can see mislocalization of mutant TDP-43, but not wild type, that there is a stress granule formation problem. There are cytoplasmic aggregates at 9 months with an age-dependent progressive motor deficit, and therefore we think this is a potential tool to study early disease and try and find some useful insights, as well as finding some drugs that might work. So this remains for me to particularly thank our funders, the M&D Association, but the back cloning was done in Richard Wade Martin's lab by Ruxandra Mutahak, who's now a postdoc with me working on stem cells. And this is about a year's work. It's really very difficult to recombine in these mutations, so that was a really very important part of the project. Ben Davis creates these mice very nicely, and most of the work you've seen on the mouse was done by David Gordon, who is a postdoc. Thank you very much.